Hello, everyone, and welcome to this new session at the BG24, the session in Genomics in the Neotropics. This is actually a very special event that we're doing here because for the first time we are putting together people sitting in the Neotropics in different countries to tell us about the big initiatives that are arising across the different countries in the region for genomics applied to biodiversity. Here we have a selection of wonderful researchers who are going to tell you about the initiatives that they are involved in or that are getting started. And today, I hope that we can put together many people sitting in the Neotropics or working in the Neotropics so that we can in the future start a dialogue and talk about the challenges and the perspectives and how all the countries in the region in Latin America and Caribbean can get together to overcome some of the major challenges which involve using genomics for biodiversity in countries in the global south. I'm going to share with you via the chat a link to a form. In this form, we're going to collect names of people who are working in the Neotropics, either sitting there, doing research there, or working with species from the Neotropics that are interested in this conversation. Later on, we're going to try to organize a call where we're going to start this dialogue. And also on top of this, we would like to ask you to help us identify people in the different countries who can potentially be leading initiatives, large initiatives of the same type that we're going to see today presented here. It could be people who are already working on that, thinking about that, or that maybe you know that would be interested. Please inform us who these people could be so that we can represent the entire region and have this dialogue really as a representative type of conversation. So I'm sending here the form via the chat, via Slido. I'm going to do this again at the end. So today I have the pleasure to present our speakers. We're going to start with Andrew Crawford and Paula Pulido Santa Cruz. Andrew is from the University of the Andes, Colombia, and Paula is from the Universidad del Rosario, also in Colombia. They're going to have a joint session or a joint presentation. Then we're going to move to Juliana Vianna from the Pontificia Universidad Católica de Chile, who is going to talk about the Mil Genomas Chile, this project that has started a while ago. We follow to Alexandre Aleixo from the Vale Institute of Technology in Brazil, who is going to tell us all about the genomics of Brazilian Biodiversity Consortium. And finally, we'll have Eduardo Eizirik from the Pontificia Católica Universidad, now I'm talking Spanish, Pontifical Catholic University of Rio Grande do Sul, also in Brazil, talking about the consortium Genotropics, about neotropical adaptive genomics. So we're going to be watching the videos, and at the very end, we'll put all the speakers together for a Q&A session. And I hope you enjoy.
Hello, everyone. I am Paola.
I'm Andrew.
Thank you for being here today. We will be presenting the biodiversity genomics in Colombia, challenges and solutions. So Colombia heads the list of mega-diverse countries, occupying 0.7% of the world's continental surface, yet it hosts 10% of the planet's biodiversity, a feature that makes it one of the countries with the highest diversity of species per unit of area on a world level. Yet, much of its biodiversity is still undiscovered. The last count was 80,000 observed species. However, these estimates remain highly uncertain, in part due to the difficulties of collecting biological samples in areas affected by conflict. Biodiversity estimates are needed, for example, to help conservation resources towards areas where biodiversity is being lost most quickly. Colombia has a privileged geographical position in the northwestern corner of Latin America, including the wet Pacific, dry Caribbean, free chains of the Andes, tropical grasslands, and the Amazonian rainforest. One of the major challenges to do biodiversity genomics in Colombia is funding. This graph shows gross domestic expenditures on research and development expressed as a percentage of GDP, and Colombia is very at the end. Other countries in Latin America seem to be spending much more money in science, and the total science budget for Colombia is projected to drop by 26% in 2025. In conclusion, we have to be creative on funding. Next step to biodiversity genomics in Colombia is permits. There are several permits for scientific research on biological diversity in Colombia. Basically, any national research institute in the academy that intend to collect specimens of wild species of biological diversity to carry out non-commercial scientific research projects must request from the competent environmental authority a permit that covers all scientific research programs carried out by researchers. But this permit does not cover communal territories. Therefore, there is also the informed prior consent, which mainly involves the dialogue with these communities to guarantee the real, timely, and effective participation of these groups in decision-making regarding projects or activities that may affect them, even though Colombia hasn't ratified the Nagoya Protocol. Indigenous groups and Afro-Colombians have experienced or are concerned about biopiracy. Genetic resources of these communities must be considered as a collective heritage. A key challenge to genomic studies in Colombia is field conditions. Collecting samples for DNA structures is complicated. The quality of these tissue samples can have a profound influence on the reliability of downstream analysis. Also, transportation is important, and it requires to maintain a cold chain, sometimes complicated by the lack of interconnected roads in several places of Colombia. So now I pass it to Andrew.
Yeah, thanks. Besides making new collections, there are biobanks, tissue collections within Colombia. This map shows some of the registered biobanks or tissue collections that we can use. It is worth highlighting, though, that only one collection is based on liquid nitrogen containers, that of the Humboldt, and each collection faces some challenges. For example, there's not so much training in making tissue collections in optimal storage, and also little awareness of special extraction methods for biodiversity genomics, for example, to get ultra high molecular weight DNA. And there's little to no funding for new freezers, for programs to systematize the data that we can share among collections, and the technical support that's really necessary to maintain these collections. So in conclusion, biodiversity genomics projects often start from zero, i.e. primary collecting, depending on the group of interest. Also, step 4 then would be generating these genomic data. We did an informal survey of some of our friends and colleagues that do genomics within the country, and we came up with this list of Oxford Nanopore, Illumina, and MGI sequencers available. Some of these have restrictions, meaning they're not generally available outside the institution that houses and maintains them. But there are options for sequencing. We were surprised to find there's NovaSeqs available, but these are run by hospitals and insurance providers, so generally not available to us as biodiversity genomicists. Okay. And
okay, so now I'm going to talk about some case studies. These within the framework of the GROW project. So the conservation genomics for management of species at risk of extinction, the Andean bear. Funding came from the GCRF UK fund. Museum whole genome sequences were generated so far to evaluate temporal changes in genetic parameters. And to measure individual identity and population structure of these species, we sequenced a reference genome, which is important because the idea was to investigate the scale of illegal wildlife trade. And so far, there are several individuals also being resequenced.
And for 2 more examples, here is the harlequin toad, which is a member of a very endangered group of amphibians. This one from Santa Marta. And this was actually funded by Pacific Biosciences, the corporation. You know, they have annual contests for sequencing genomes. So we won one of those. That required a few permits, not too hard to get. And the data, including export, the data were then produced at BYU. And so the assembly was all PacBio, of course, and we got a really good final result. And for the last example then of biodiversity genomics in Colombia, there are other studies. We're just bringing you 4 examples. This one was generated again at Rockefeller as part of the Vertebrate Genomes project. So where they generated a chromosome level assembly. And then in Colombia, also in Manhattan, the Endofato lab generated low coverage resequencing data, and all that with the goal of discovering the genetic basis of this color pattern polymorphism that I'm showing you in the figure. And so possible solutions then to these challenges. Our previous examples focused on sequencing outside, which has been a great success. It would be nice to sequence within country. We've mentioned some of the platforms. This would reduce export permits and cold chain issues. So this is an exciting direction that we hope we can work with Oxford Nanopore and other options. Computational resources, we didn't talk a lot about, but they exist in Colombia, but they are limited in terms of access and time that you can get. The Galaxy model provides a great alternative to many of us working in Colombia for doing our informatics. And we require access to more parts of the country. We need to fully implement the peace accords. And we're very excited by the COP16 going on right now in Cali, Colombia, where important topics include technology transfer and other partnerships between say developed and this country. And so we'd like to talk later then about possible regional node within South America or Latin America, and Colombia has a lot to contribute to this, but also some challenges that we've mentioned. Some of the strengths of working in Colombia are the great human resources we have. We have very smart people, wonderful students, highly trained people, successful researchers. We have tremendous biodiversity, which Paula emphasized already. The tissue collections are growing, but they need a little support. We have some sequencing platforms available, not PacBio, but others. Permits are relatively easy, and I think there's a growing interest in from wildlife authorities in supporting biodiversity genomics. Some of the struggles we've already mentioned, including national funding is very low. Some of the country is still not accessible, and no PacBio, computation limited, and biobanks we hope are going to be growing soon.
Thank you.
Thank you for your attention.
Hello, everyone. I'm going to present about the 1000 Genomes Chile. It's an initiative to generate reference genome for the Chilean biodiversity. So we are composed by different centers of excellence, as is known in Chile. Those are long-term projects composed by different researchers from different areas, different university institutions. And I'm part of a Millennium Institute Center of Genomic Regulation, and this center is leading this initiative. So we are also part of Earth Biogenome Project, and also we have been supported by different members of the European Reference Genome Atlas, such as Dr. Camila Mazzoni, who came for our workshop this year, our first workshop. And this picture is from the first day of the workshop. We even had more people in the second day, which was more technical discussion. And for next year in April, we're going to have our second workshop, which we have a pleasure to have a talk of Dr. Harris Lewin from EBP. So I'm the chair of 1000 Genomes Project. We have a communication director. We have hired a data manager, and we have a different committee in species selection and sampling, sequencing, bioinformatics, knowledge transfer, ethics, and legal and education. For example, we're starting the debate about legal protection of genomic data of our biodiversity. So in the phase 1, we're going to sequence a reference genome for one genome for each species, the novel genome. And the phase 2, we're going to do resequencing to capture local adaptation to this large, wide latitudinal distribution from the desert forest to southern Chile and Antarctica, such as doing other initiatives as in California. So you already know why to sequence genomes and the importance of that, but we have these 4 main action areas and studies to show the society, scientific community, private companies, and the government the importance of this 1,000 Genome Initiative. For example, genome function and evolutionary history. In Chile, we don't have this huge biodiversity as the tropics, but we have high endemisms because of the isolation by the Andes and the desert and the ocean. For example, we have up to 65% of endemism in reptiles and amphibians. We have up to 50% of endemism in plants. So we want to understand the adaptation to extreme environments as we have in Chile, the high Andes mountain, desert, going to the Patagonia forest and up to Antarctica, and also the deep sea. So this is very important regarding we are facing the climate change. In the case of Chile, for example, we have the desertification, it's expanding further south. And here in this study, we try to understand the adaptation of guanacos to the desert, but also to the high Andes and to the southern Chile. We also sequence genomes of penguins and to understand adaptation to cold water, but also how the environment has implications in the speciation and diversification of this group. So the second area is bioeconomy. This is the white strawberry found in Chile. It's native from Chile. But we have also a high diversity of marine organisms that are used for consumption. They are distributed in upwelling system of rich in nutrients which support all this diversity. And we have kelps as well that are used in pharmaceuticals. Another area is the public health. For example, we had in Chile an outbreak of highly pathogenic avian influenza A, starting from the north to south, and this killed 15,000 seabirds mostly, and then jumped to mammal. We know the implications of that, killing 3,000 sea lions. And last summer, we did a survey in Antarctica, and we found some cases of this highly pathogenic virus in an Antarctic peninsula. So it's the genomes can contribute to understand which species are more resistant or more susceptible to this kind of virus, and the genes involved to this resistance or susceptibility, and also this can contribute to the implications of management in new areas such as Antarctica. What is going to happen? So the next one is conservation of biodiversity. And this study was a collaboration with Dr. Eduardo Eizirik here. And here you have the marine otter and southern river otter, Lontra felina and Lontra provocax, found in Chile. These two species, Lontra provocax and Lontra felina, has the lowest genomic diversity of all otters in the world. So when you compare, for example, the genomic diversity of a southern river otter with the neotropical otter, you can see which is this low diversity is distributed across the genome. We know the conservation implications of that, the low ability to adapt to environmental changes, and we also there is other studies also supporting this low diversity for other species in the southern Chile, not only southern river otter, but for example, the Darwin fox distributed in this area comparing to all other Lycalopex fox, and also this small feline guinea, which has really low genomic diversity compared to all other Leopardus felines. So this is the workflow from our project. And for sampling and taxonomy, we have a panel of researchers for different taxonomic groups, and they are building a long list of species based on these criteria: scientific question, a taxonomic and geographic representation, bioeconomic importance, cultural significance. And this is uploaded in GOAT, and they are also constructing a shorter list of priority to sequence, and part of this species goes to public election. So the public election has a special goal of outreach, public engagement, education, and use outlets. So these are the first group of species that was submitted to voting to the society for first just vertebrates. And the people could go into the web page, read about the species, their distribution, and the conservation status. And then they could go to the election of the species, and the poodle deer, the small deer, won the election. And it was all over the news, which has a special importance for conservation and know a little bit more about this endemic species. So, and now the election is for plants. You can go to our web page and see which species are winning. Now it's the cactus from the north is the one that is going first. And the second step is, of course, sequencing. We are using a combination of methodology, including long read sequencing. We don't have PacBio in Chile, but we are using nanopore and Illumina. Our goal is to sequence the genomes in Chile, contributing to all the externalities such as formation of human capacity, social and economic implication, impact, and public health. So the last step is the workflow from the sequence to assembly and annotation. We are also doing transcriptomes for annotation, and we already have a GitHub for our project. All these data will be analyzed in a national high-performance computer placed in Santiago, and now the Chilean government gave money to place a bigger computer in the north of Chile. So we also have a work with school students. We sequence these small invertebrates, and there was a publication with students using this genome data. And last year we went to the north of Chile, in San Pedro de Atacama, and we have been also to the southern and other schools along the Chile. And next we are going to the eastern island. So we have all these genomes already sequenced or being sequenced and assembled. And the goal of our project is to sequence a wide taxonomic representation. And we have this ambitious number of 1,000 genomes to contribute to the bigger project that is the EBP, which is even bigger, more ambitious, to sequence the entire world biodiversity. Thanks for listening. Thanks for the organization of this conference. And if you want to know a little bit more about our project, you can go to this QR code. Thank you very much.
Hi, everyone. Wish to thank Camila for organizing this roundtable discussion, and also to the other colleagues that are in the same session with me. So I want to talk to you briefly about the genomics of the Brazilian Biodiversity Consortium, which is hosted by ITV, Instituto Tecnológico Vale, in Belém, Brazil. And ITV is a multidisciplinary research institute, and one of the research hubs there deals with genomics, environmental genomics to be more specific. So That's where the GBB, as we call it, is posted. A little bit about the GBB structure and governance. So GBB is the management of the GBB is shared by ITV and ICMBio, the National Brazilian Authority for Biodiversity Conservation. And GBB has two major hubs, an environmental DNA hub and a genomics hub. The environmental DNA hub is divided into two sub-hubs, the barcode reference, which aims to generate super barcodes for Brazilian species, and the meta-barcoding sub-hub, which aims to implement meta-barcoding survey methods and protocols in the different Brazilian biomes. Then two sub-hubs under genomics, the conservation sub-hub, which essentially will use reference genomes, will produce actually reference genomes and resequence genomes for endangered species. And then the bioeconomy sub-hub, which will produce reference genomes and resequence genomes for species of bioeconomic interests. Of course, to support all these efforts, we have a very strong support from our IT team and also from our data manager team, which will deal with the meta data associated with the different samples that we include in the analysis. GBB is fully funded by the company, by the Vale company, and we have $25 million until the end of 2027 at least. And since October 2023, so 1 year from now, we have been affiliated to the Earth Biogenome project. How are species defined as, how are they elected to be included in the project? So essentially, ICMBio chooses which species will be sequenced. And, but of course, they base their decision on current evidence, mainly the national action plans for the conservation of different species, but also internal consultations. And we have had so far two dedicated workshops to consolidate this list of taxa that are gonna be sequenced. So essentially, I'm talking here about endangered species, which are already listed as endangered, and species of bioeconomic interest. What are GBB goals? To generate at least 80 high-quality reference genomes at the chromosomal level, over 1,000 resequenced genomes, about 1,600 super barcodes, and 1,600 environmental samples from major Brazilian biomes. The current status of these different targets. So far we have implemented projects concerning a total of 50 reference genomes. So we have 30 more projects to go. And the way we organize these projects is that we consult with ICMBio and other researchers throughout the country who are interested in generating genomes for their species of interest. So whenever all our interests match, then there are genomes, down the line. So we are still looking for 30 more genomes to do, and but this is well on track, and we should have that number soon. Hopefully by the end of the year, if not by the middle of the middle part of next year. And about so far 40 species are being targeted for the population genomics, which will generate a lot of resequenced genomes. And we have already generated 1,329 super barcodes. So I'm talking here about organelle genomes. And so far we have collected 614 environmental samples from the Amazon, which are already being sequenced as I speak to you now. We are already producing papers and generating scientific output from the genome. So this is the first GBB genome published together with the VGP project, which was in fact generated before we had our sequencing part fully in full operation. There are many more species down the line, so we are already working on genomes for these for all these beautiful species there. And this is our first reference genome generated fully at ITV for the sterile for Docnemis expansa, an Amazonian, a widely distributed Amazonian turtle that's also exploited commercially and also as food by local populations in the Amazon. The workshops, we have carried out several workshops towards capacity building of ICMBio personnel mainly. In this case, I'm showing you pictures from the eDNA metabarcoding survey training that we had last year. And this year in June, we carried out the first eDNA sampling at the Floresta Nacional do Tapajós, which is located near Santarém at the heart of the Amazon. And we should have the data already available from this survey by the end of the year. And it was a great teamwork together with ICMBio. And this is the first time that eDNA was paired up with the efforts that ICMBio conducts at this site using standard survey method. So the next steps to compare what the eDNA and the sort of standard field surveys deliver in terms of species detection. So if you want to know more about GBB or still actively looking for partners both in Brazil and elsewhere, please check our website and drop a line. It will be great to connect. Thank you very much.
All right, so hello everybody. My talk is going to be about the Genotropics Consortium. It's a consortium of researchers that focus on several kinds of research addressing neotropical adaptive genomics and other related topics. It's a consortium of Brazilian researchers, a Portuguese researcher, including different institutions. And so today I'm going to give you an overview of what this consortium has been doing and what its proposed strategies are for the coming years. So the group was founded by 5 researchers that are based in Brazil. These 3, a researcher from Portugal who's based in Germany, and a Brazilian researcher who's based in Germany as well. This is the core group, the founding group. It was funded initially, and this is the initial funding period, by both Brazilian and a German governmental agencies. So it's an exchange and academic partnership program that has as its core the idea of investigating biodiversity genomics and connecting scientists working on biodiversity genomics in Brazil. And our website is here. So throughout the talk, I'm going to show you the website, and you guys can go visit later on to get more details on it. Right. So the initial composition of the network was 5 PIs, professors from 3 Brazilian universities who are placed here, the universities that I showed in the previous slide, UFMG, Unicamp, and PUC-RS. These are the locations of these 3 universities. And there are 2 PIs based in Germany, Camila, who's Brazilian, and Ricardo, who's Portuguese, both based at German universities and museums. There are 2 associated PIs from this university and that, who also run groups focused on biodiversity genomics, and 2 postdoctoral fellows who are Brazilian and based in Germany at this point. Then early this year, in 2024, we performed an expansion of the consortium by adding 5 additional PIs who were interested in joining and who also perform biodiversity genomics research. And the location of their universities, these are 4 additional universities located here where the yellow dots are. So this is the current structure of the consortium. So what's the idea? What are we trying to do? So it has 3 pillars. One is scientific articulation, the other is capacity building, and the other is stakeholder connection. The vision and mission of the consortium are shown on this slide. So the vision is to reach a deep understanding about species adaptive processes in neotropical biomes across time, including adaptive capacity of species to environmental changes. And the mission is to utilize and promote the use of state-of-the-art genomics and large-scale analysis of adaptive processes in neotropical species, including analysis of evolutionary history associated with environmental adaptation. So these are the general ideas that give us direction towards which we're trying to move. So these are some specific objectives that we have in the Genotropics Consortium. So one is to connect experts in the region working on genomics. The other is to train students and young researchers in the latest and most advanced genomics techniques that are available. The other is to elaborate, to produce new grant proposals, and we've been doing that in the last few years, just trying to get more funding to generate data and to analyze data in genomics. The other is to analyze high-quality genomes using comparative approaches and population genomics. The other is to generate new genomic data sets that represent intra- and interspecific divergence and environmental adaptation. We've also been pursuing that with several species, as I'm going to show soon. And the other is to produce protocols of legal and technical aspects to guarantee data quality and equity in the exchange of samples and genomic data. So these are the objectives that we try to pursue within the Genotropics Consortium. So within the scientific articulation pillar, we've already been performing various collaborative projects. Our idea is to not only pursue projects that we're already doing in collaboration among the PIs that compose the consortium, but also to produce new ideas and new projects and to bring in more people to collaborate in various aspects of biodiversity genomics. So these are some of the taxa that we have been studying, as partnerships and collaborations among the PIs in the Genotropics Consortium. So it includes vertebrates so far. So most of the projects that we do are on vertebrates, includes aquatic and terrestrial vertebrates, vertebrates from different biomes in Brazil and other neotropical countries, with a variety of questions. So this schematic shows some of the questions that we try to address. So questions related to adaptation, I'm not going to read all of them. Questions related to comparative genomics, questions related to phylogeography and to speciation. So basically, to really reach the point where we can study adaptation in depth, we need a lot of other baseline information on the evolutionary history of species, including, for example, these topics here on the right, and these topics here in the middle. So basically, we need to understand species biology and evolutionary history, and then move towards adaptive genomics questions, which is something we have already been accomplishing for some taxa, but for others, we're just in the infancy of understanding their basic biology and evolutionary history. And then there are some applications, right? We can apply those knowledge advances that we hopefully will promote to a larger degree in different ways. So understanding ecosystems and connecting ecosystems to a variety of aspects of societal interfaces, such as sustainable development, food security, and others. Most of what a lot of us focus on is conservation related. So understanding species as a basis for designing and implementing conservation strategies, and also applications related to societal health and societal well-being. So there are many topics that we'd like to help develop using genomic technologies. Regarding the capacity building pillar, the idea is to use different resources, different formats to promote training through workshops and other shorter activities, online activities. We've done one in 2023 in Belo Horizonte at one of the participating universities in Brazil. It was a hybrid workshop. Then we did another hybrid workshop, mostly online, but with some people in person at Campinas, in Campinas at the Unicamp University. This was early in March this year. And next year, we'll have the workshop focused on population genomics. The previous ones were focused on these other topics, genomic methods in non-model organisms and egalitarian collaborations for the future. A lot of these are online in YouTube, and you can actually watch the presentations and see the discussions. A lot of it was in Portuguese because we want to promote engagement of young Brazilian students, but some of the discussions are in English. And next year, it will be held at my university here in southern Brazil, PUC-RS, focused on a hands-on training effort for population genomics. This is going to happen next year. This is a picture, a screenshot from the 2023 workshop with 60 participants from all over Brazil. Some people were in person and several others were online. And it was a big success and a lot of engagement emerged. We have a lot of people signing up to our mailing list and interested in learning techniques and applying genomics to their specific systems. With respect to our third pillar, the stakeholder interconnection pillar, we'd like to promote scientific partnerships that expand the number and scope of the projects within Genotropics, but also with other networks and other consortia. So partnerships with other networks with complementary objectives, and there are others in Brazil, including here in this session, we'll be hearing about another large consortium that is pursuing very complementary goals. Partnerships with actors that may use the genomic knowledge for applications in biodiversity and societal interfaces, and additional partnerships with government agencies, NGOs, companies that may support the broadening of capacity building in biodiversity genomics. Our idea is to move towards a broader network, a network of all the consortia and all the initiatives that would like to help promote biodiversity genomics in Brazil. So we're trying to construct, Genotropics is one of the players, along with other stakeholders, other networks. We're trying to build something that's going to be called something like Brazilian Network of Biodiversity Genomics that integrates generation of data of various kinds, whole genomes, resequencing of genomes, DNA barcodes connected to whole genomes, SNP panels for a variety of applications in conservation and other areas, monitoring of species diversity, genetic diversity, applications in conservation and sustainable use, research and innovation. So the idea is to use biodiversity genomics in a variety of ways, to be integrated with international efforts such as EBP, and to create capacity building and momentum in Brazil for biodiversity genomics. So the idea is to use platforms such as this session to promote such integration and complementarity and synergy. And I look forward to the discussions later today and tomorrow in our additional Zoom session. Thank you very much.
All right. Yes, thank you so much. So exciting news coming from the different presentations. And we can see across the different countries also different stages and that the projects are, or even some future perspectives from Colombia and projects that are already, well, that even have been relaunched, such as the 1,000 Genomes Chile, and rebranded. So that's now hopefully it's going to be to take off. And the GBB in Brazil already full on in production. and the Genotropics being a consortium of different scientists pursuing the same or similar goals. So I think we have a couple of questions here in the Slido, and I would like to use them also to guide our discussion a little bit. So maybe we can start by talking about some of the challenges that we have in Latin America and Caribbean, even though here we are basically representing South America, we can say, in this particular call. But I think the challenges are going to be similar all over. And so let me go to a question that was Christopher Laumer was saying that he's heard that it can be very difficult for Latin American researchers to get a hold of nanopore flow cells and sequencing kits. And when they are available, they're more expensive than they would be for European or US researchers. Is this generally your experience? I think we all know the simple question, yes, but would you guys like to elaborate a little bit on that and maybe expand that? So not only for the flow cells, but in general, what is the difficulty in using technologies in your countries? Who would like to start?
So I can start. Fortunately, there is a couple of years we have a company that represents nanopore in Chile that makes much easier to have everything imported and buy it here. Of course, as we are importing products, there is taxes, and the local company also charge for these products, like it has an extra cost. Fortunately, the owner of the company in Chile is a big supporter of our project, so it's helping us a lot. Also, to several meetings with nanopore in direct to nanopore. But of course, there is like taxes and other things that makes much more expensive, but much more easier that this company can import everything and be responsible for all that we get the products like in our lab in a good way. So this makes much easier.
Yeah, Alexandre, I think you have also experience with oxonanopore in Brazil.
Yes, I should say that what they put in the chat is really correct. That's our experience too. However, things are changing very fast. We had a conversation today with some people from the sales management team from Nanopore, and they are completely restructuring their Latin America sales group. And it was really fortunate to hear from them that they are really interested in tackling our problems. And I mentioned to them that I see Nanopore as a main technology because of its reduced cost, and also it delivers quite a lot for not only for reference genomes, but for barcoding, for eDNA, if you want. So I think things are changing, and they are hiring. Actually, it was very fortunate as well to be talking to women in Nanopore who were Latin Americans too. So they are really realizing that there is a huge potential that has been so far untapped by the company.
Okay, well, I think these are excellent news that companies such as oxonanopore are already looking with, let's say, special eyes at the region, because there are peculiarities and there are struggles that the global south doesn't share with the global north. And this is something that the companies should really be watching and helping the region to find solutions for that. So it's great news that there is, some companies already going towards this. I will maybe switch here to another question so that I can tackle Eduardo, Paula, and Andrew. And this is from Daniel Foster. He was asking, so can you say something about translating conservation genomics results into conservation actions in your regions? Would you like to start? Go ahead, Eduardo. Oh, yeah.
Can you guys hear me all right?
Yeah.
So, well, first of all, I love the other talks. I think it was really nice, good session with lots of interesting stuff. I think we've been trying to do exactly that, to use some of our genomics results to empower conservation strategies, especially on behalf of carnivores. The species for which this is most advanced is the jaguar. And so next week, for example, there will be a workshop held by the Brazilian government on jaguar conservation, which will present the current genomic status of the data, which will be used to direct conservation actions, both in the wild and in captivity ex situ. So basically, the management of jaguars in Brazil is currently directed by the genomics results that we've been obtaining. So I think in this case, it's directly applied, and we've been developing markers for faster and cheaper typing of jaguars for multiple uses based on the genomes we generated. So both the whole genome data, such as runs of homozygosity and other features, but also markers developed from the genomes for quicker typing. That's, I would say, a direct application for conservation.
And Eduardo, could you maybe just briefly mention what is the acceptance level from the government side on this type of data? Do you think this is something that they understand already, they accept, or there is still a road to follow?
My impression is that it varies considerably across taxa, and I think Alexandre will have an even broader perspective because of the interaction with ICMBio and the GBB project. In my case, it's perfect. The interaction is very smooth because we've been working together for decades before we started doing genomics, and so it is a very smooth and collaborative relationship between the carnivoran oriented managers in the federal government and state level as well, and us doing genetics and genomics. But that's because we built this relationship over a long time. It's not necessarily the case in every taxon. Perhaps Alexandre, looking at the broader panel of taxa, can complement, but I would say it varies. In some taxa, it's very smooth. In others, it's beginning. In others, there's no connection yet. It's just starting. That's my impression.
Let me then check with Colombia. So what is your experience, Andrea and Paula?
Yeah.
I think Paula should take that one. Go, Paula.
Yeah, sure. Well, we have few cases. I think we still are working on several projects, and the data is being analyzed. And sometimes it's complicated that we don't have the right amount of data to target a conservation question. So for the emblematic Andean bear, for example, we do have several museum genomes, and that has helped us to understand, for instance, or evaluate temporal changes in the genetic diversity, for instance. So then we can target some areas or geographic locations or places in Colombia where we have to implement some bigger effort for conservation, mainly in the northeastern part of Colombia, close to Venezuela, for instance. So that was an impressive finding, I believe, with a big amount of genomic data, because sometimes you see other results with a few markers that have conflicting results, and when you get the genomic whole picture, so it is clearer. And I believe what it was interesting in this work is that we built a very close collaboration with regional environmental authorities, national parks, and of course the universities, so to try to put all the information that we got in the National Conservation Program for the species. So we are working on it. It's a work in progress, but that's one of the examples. And we are trying to do similar conservation strategies for species that are quite endangered due to illegal wildlife trafficking. So, but we are waiting for the results. So, yeah.
Thanks, Paula. So I actually wanted to point out to the audience to please look into the link that is in the chat for the cognitive forms, so that you can put your name in case you're interested in following a list of communication on genomics in the Neotropics, or if you can suggest maybe initiatives that are starting the different projects, or potentially leading people that would be interested in starting and have a dialogue with other countries on the topic. And then I will go to next question here, which was from Catarina Torres, asking Juliana and Alexandre, who have the projects running, what are the policy for public availability of the genomes being produced in your projects? If you can say in few words, because we don't have much more time, I would appreciate.
Yeah, very short. So every genome that we are sequencing, there is a researcher leading this with an important scientific question behind. So he's responsible to coordinate all the other teams, bioinformaticians and everything. And of course, publishing, we have embargo time before this embargo time. And when he's published, all the genome will be available, otherwise in the embargo date. That was short enough.
Yes, thank you. Alexandre?
I will also rush, and I think we also have a data sharing policy that's being implemented, and the idea is to put all the genomes in NCBI and GenomeArk, since we are an EBP hub, as soon as they are assembled and curated. But there will be an embargo there that's going to vary depending on the genome. So we are following the same method such as like VGP. So no surprise there.
Okay. So here, that's an interesting question. Yvonne Giska is asking if there are international initiatives that are sequencing species across borders from the Amazon biome or from the Andes. And that's an excellent question. There isn't anything that I guess we are aware of, unless you guys are aware that's something that cross borders. But that's exactly what this type of dialogue among countries could bring, right? So that's even another instance. Do you guys know of anything in this direction?
Yeah,
I just wrote in saying that that's what they try to promote.
Yes.
We are discussing that with Argentina. We have had some meetings already, and so that's in the pipeline.
Okay. Let me see here. Cristobal is asking if environmental DNA has been used to identify presence of endangered or elusive species. It's the eDNA. I don't know if your projects touch that. I think Andrew uses eDNA, the GBB as well. Have you had any success on detecting endangered species or elusive species using this type of data?
We are looking now, and we don't have the results yet, but it's in the works trying to find lost frogs using eDNA and streams from the Andes. So hopefully by Christmas we'll have some news for you.
Awesome.
But good question. Thank you.
We are on the exact same page as Colombia, just waiting for the results. But we are targeting specifically vertebrates in general and macrophytes in another project.
Okay,
Yeah.
so we don't have any more time. I just wanted to ask you guys, do you have any final words? Like, why do you think the Neotropics should get together? And I mean, in 1 or 2 sentences for genomics for biodiversity. And let's start now with Juliana.
So yeah, first of all, we have a huge biodiversity, lots of endemic species. We have to coordinate more between countries, as what was the question, and also build capacity in the area. So that's very important to promote, like teaching new generations of future scientists in genomics.
Thanks. Paola, 1 or 2 sentences.
Sure. Yeah. Well, I believe that this is a great opportunity to also really innovate methods, steer experimental design, produce and analyze data in a very collaborative and nice network, and hopefully we get funded research projects.
Eduardo?
I agree with them.
Alexandre?
If we don't do it, nobody will do it. So let's do genomics in Latin America.
And Andrew, so that we can close.
Yeah, I think of all the Colombian PhDs who are not finding jobs in Colombia. And so I think we need to support them. We need to support biodiversity here,
Yeah.
and each country and the region needs to make their decisions.
Thank you, everyone.